All right. So I'm jumping right in. So Hilal did give me a title. I decided that it made no sense at all. So I came up with my own title. But then Hilal also said to me that he wanted me to weave in some discussion of phase separation, because I think this is a theme that honestly has been completely missing from today's discussions. So I will basically split my talk into two parts. One will be sort of some concepts around phase separation that are emerging, and then sort of pivot toward some work that I think might be relevant from the HD standpoint. Okay, so essentially a theme that has emerged in all these discussions about complexity is that things happen at the right place at the right time in cells, right? And they happen so in cell-specific ways. And I think there's been a sort of search for an organizing principle for how things happen at the right place and the right time and how that might go wrong. And what I'm going to try to say, and you can challenge me all you want, is that it appears that cellular processes are controlled by macromolecular phase transitions, such that disturbances in these types of phase transitions, particularly in post-mitotic cells, might well be sort of relevant to the driving of degenerative cues. So what I will do is quickly brush you up on the concept that has emerged over the past few years of biomolecular condensates. For ever and a half, we've recognized that there are lots of these membraneless bodies that actually are present inside cells, particularly in the nucleus, but also in the cytoplasm. About 12 years ago, there was this idea that macromolecular phase separation might in fact be the underlying process by which these types of membraneless bodies come about. And that gave rise to, I think, a bit of a rushed convergence upon liquid-liquid phase separation as the theme, right? And it kind of became a thing to say liquid's good, solid's bad, as with all simple things that turns out to be absolute balderdash, actually. So it turns out, of course, that one theme that we all agree on is that macromolecules need to be multivalent in order to be able to drive phase transitions. But multivalent in what exactly? And it turns out you can think about multivalence from the language of associative polymers of what are called stickers. And these are basically entities that enable the formation of reversible physical crosslinks, much like you would make a gel in precisely this manner, right? Have lots of reactive groups that can chemically be cross-linked. In this case, nature is giving us these stickers, either in the form of amino acids or in the form of structural motifs that can be physically cross-linked. Dating back all the way to the 1940s, Florey actually sort of clarified for us exactly what would come about when we think about physical or any kind of cross-links. So these are specific sticker cross-links, and typically what will happen is that if a particular molecule has got enough of these Pac-Men on them, the idea is that there's enough of them so they can start to sort of hook up with multiple molecules to make a network. And so concentration becomes the operative order parameter, whereby if some threshold concentration is crossed, then you make a system spanning network. So all of us are bivalent. Let's just pretend that we were trivalent. We had 3 arms. That's the basic requirement. We start joining arms. Very much, if this room was a little more concentrated, we could essentially have a network that spanned from one end of the room to the other that would have some characteristic topological properties. That is essentially a gel, right? But it turns out that we can solve the problem more simply if all of you were to huddle up here. So we create a high local concentration. We take advantage of our multivalence, and we basically now make a small volume spanning network. And that would essentially give rise to coexisting dense and dilute phases, whereby there is some threshold concentration defined by the solubility of the polymer that's crossed, and we end up with a droplet that's basically system spanning or droplet spanning. So the idea then is that this has been cut off, but the percolation threshold is somewhere between the sort of threshold concentration for the density transition for huddling all the molecules together, and then inside of that condensate or the droplet we cross the gel point, so to speak. So condensates, therefore, are not boring Newtonian liquids, right? They are, by definition, viscoelastic materials that have an underlying topological structure. So the underlying structural motifs or the chemistry will determine what sort of networks are formed. And that means that if there's one fundamental message you take home today from this particular talk, it is that basically a condensate inside of a cell can span a spectrum of properties from being purely viscous to purely elastic, depending on the types of crosslinks that can form. So what will matter is the sticker to spacer ratio, let's say, within a particular sequence. Something like polyglutamine will just be polysticker. Something like polyserine will be polyspacer. And of course, we've got a whole bunch of cocktail in between, right? Now, things like when we say liquid, we only focus on interfacial tension, but in fact, when we start to move toward more viscoelastic materials, we start to focus on the structure of the interface. But here is the really important point, which has been cut off. The material properties of a condensate depend on at what time point we interrogate them. Right? Because essentially what ends up happening is that condensates that are viscoelastic materials exhibit what is called shear thickening phenomenon. They'll also exhibit shear thinning, but you need to burn energy to do that. That means that the viscosity of these materials will increase as a function of time because you have a whole bunch of crosslinks that are forming and breaking. So those internal dynamics really matter. And so simply saying liquids, or liquids good, solids bad, a really, really bad idea, right? So essentially then what this means is that there are a whole cocktail of phase transitions that give rise to a whole bunch of complex phases depending on cellular state, cellular location, cellular age, and the interplay of homotypic versus heterotypic interactions. Here's simply a montage of different phases one can think about. I've talked about phase separation coupled to percolation. This is an active emulsion, so essentially the cell is burning energy in order to be able to maintain condensates of a particular size. But there are fibrillar solids. We've talked about those endlessly. But what I will talk about is another class of phase transitions called microphase separation, or really micellization. If a molecule has a surfactant-like structure to it, then what it will do is essentially make what looks like a microphase. That's my pivot to actually Huntington's disease, which will surprise you, but that's okay. But essentially, years ago, I got interested in this problem because I thought, okay, if all the neurodegenerative disorders, this should be the tractable one. It's a monogenic disorder, right? One gene, one disease. My goodness, we should be able to get this right. And here I am standing in front of you. Either I'm terribly stupid, which is certainly a real possibility, or it turns out that there are also complexities that we've all ignored. So one gene, one protein, but multiple protein fragments give rise to complexities on all kinds of scales. So the first thing to think about is let's only focus on the exon 1 spanning region of Huntington. It's got this amphipathic region, highly conserved for a 17 residue stretch, impressively so. It's got this polymorphic polyglutamine region and a polymorphic proline-rich region, right? So this is basically a block copolymer, if you really want to sort of coarse grain it. And in fact, a few years ago, Hilal, Edward Lemke, and I teamed up to basically say, let's try to solve the problem of the structure of monomeric Huntington exon 1. Because is there or is there not a pathological conformational conversion? The answer turned out to be no, but instead what you actually end up with is basically conformations that look like this. This is sort of atomistic simulations integrating a lot of data from experiments on the single molecule variety. And essentially what you're seeing here is a conformationally heterogeneous tadpole, right? Basically, the overall coarse-grained architecture really looks just like a piece of sperm in that it's got a head, it's got a tail, and that head in this case is a sticky head, right? So it looks like a surfactant, but in the opposite architecture. What that does is it generates morphologies that look like inverse micelles, right? And so you get this microphase separation. So in fact, what we've been able to measure in the test tube is that we get 3 distinct phases defined by two distinct critical micelle concentrations. The formation of an S phase, very well-defined sizes and shapes, and then, of course, the fibrils that happen at higher concentrations. This looks just like the phase diagram of a micellar system. And in fact, here are sort of TEM images of the S phase versus the F phase. And what's beautiful about this is that if you can actually go back and look in sort of the R62 mice, you see these spheroidal structures, something that Steve Finkbeiner showed us back in 2004 as being the relevant markers as far as toxicity was concerned. And these are data from Jill Bates's lab. So a few rules that have emerged. These threshold concentrations, and here is a really important point. The director of NIA is here, so I can say this. NIH study sections do not like it when people measure concentrations. They say that's very boring. I tell you something. From a physics standpoint, you don't measure concentrations, you're not fucking doing anything, right? Because basically, without that, there's no physical chemistry you can define. So basically, what happens here is these threshold concentrations decrease with polyQ length because, as Peter will tell you, it's, chemical potentials, right? You have essentially sort of threshold concentrations that change with polyQ length. This N17, it turns out, actually sort of pushes systems towards fibrils, whereas the proline-rich region is pushing things toward more spheroidal architectures. But what I want to do is very quickly, I don't know who's keeping time, Jose is excellent. So we'll connect structure to gain a function here. Oh, excellent. So essentially the idea then is, how do you think about toxic gain of function? This sticky lollipop becomes more prominent as we increase the polyQ length. Essentially, it's a multivalence argument again. You have lots of weak interactions that are enabled through the polyQ region. And so just as a proof of concept, we teamed up with Danny Hatters, who basically said, let's do a series of transient transfections in a particular cell line. And what you find is that you start seeing that there's a gain of function interaction with a lot of proteins, FUS in particular, which is supposedly a nuclear protein, but during stress will show up in the cytosol. And you'll see quite a few of these proteins popping up. They're actually drivers of stress granules, but they show up as wanting to interact with Huntington, right? So what that says is that there's a combination of heterotypic interactions, a combination of homotypic interactions, all of which gain prominence continuously, not some sharp threshold, but continuously as we increase the polyQ length. There is one last theme I'll hit because I don't think I'll have time for the last theme. So here is another issue that this is something we stumbled upon recently. So the pathological hallmarks of Huntington's disease, and Hilal's worked on this problem as well, are neuronal inclusions of expanded polyQ containing Huntington exon 1. But there's a very interesting result, which is, the question is, what do Huntington fragments actually do inside the nucleus? To date, what we've known is that if really long polyQ containing molecules make their way into the nucleus, cells die. But these are in petri dishes, right? There's also a result, and here the issue of the appropriate model system becomes a major focus. A few years ago, 2 separate groups showed that if you append N17 to GFP, then you end up with a nuclear export signal. Things go out of the nucleus. Except that is N17 appended to GFP, right? There's no Huntington there. It's that you're studying N17 pathology, not Huntington pathology. So we turn to the oocyte system because the nuclei are so big you can see them from Mars. So essentially, we figured let's sort of use this as a system that we can sort of prototype cellular localization. So here are a few constructs. This is wild type. This is a delta N17 popularized by William Yang, and then a Q46 containing one. And the idea is fairly straightforward. Irrespective of whether N17 is on there or not, the Huntington constructs have no problems going into the nucleus, and this is true even for wild type. But the really fascinating result, and this becomes the source of the question I asked Judith earlier, is that the Huntington molecules are occluded from the nucleolus. If we put a nucleolar localization signal onto them, the cells basically die, right? I mean, so essentially, so inside of the nucleus, there's still enough of a barcoding that says that you shan't go in here. And what does that do? That starts to impair ribosomal production, right? So essentially, this is a really important problem because what it's saying is that there's this occlusion from the nucleolus, but it happens to make its way into the speckles, right? So you can corrupt the speckles that way. So I think I'm out of time, or I am out of time. So I'll stop there. But if you ask me questions, I'll tell you about what we've learned about ligands sort of modulating Huntington phase behavior.
Thank you, Rohit. We have time for a couple questions. Dan?
Oh, yeah.
Oh, I take your microphone.
Just wondering, so there are like what, 9 poly-Q diseases. Does this work for all of them, or
Does what work for all of them?
this picture with the N17? How specific is this just to Huntington protein? Does it work for the other?
N17 is only on Huntington. It's
Okay.
only conserved in Huntington, right?
Okay.
And so, of course, the poly-Q length dependence is very poly-Q-opathy dependent as well. So the sequence context absolutely brings a lot to the table for each polyglutamine-opathy.
Okay.
Yeah.
So Rohit, how do ligands modulate the behavior of HTEX1?
Thank you. I'll give you your money later. So essentially, very quickly, what I'll do is point out that, oopsie, what did I just do? So the particular concept that we have sort of uncovered recently is this old concept of polyphasic linkage that Wyman and Gill sort of put together, right? Which simply says that threshold concentrations for phase separation are dependent on the preferential binding of the ligand to the two phases that are coexisting in equilibria. And so the particular problem that we sort of solved was profilin, an abundant protein that binds to polyproline, actually sort of facilitates the disaggregation of Huntington by preferential binding to the M phase through a combination of binding to profilin, which is additive, and then these weak multivalent interactions with the poly-Q region, so it becomes more efficacious with the longer poly-Q tracts, right? And so that's a concept that's actually can be used to design ligands to push the equilibria in a direction that you want. But what's interesting about that is, as Nacho will tell you, there are lots of interactomes. So you cannot just take one ligand and push the reaction in a direction that you want. You have to now remember that there are other ligands that are competing with the ligand that you're trying to sort of oomph up. And so one of the things that we've started to do is actually take on this problem of what we call a small-scale systems biology problem of asking how ligands that pull in different directions impact the underlying phase equilibrium. Right? So.
Okay, thank you. So while Peter gets set, there's another fast question.
Okay. Oh, Jeff.
Jeff, I let Jeff one more question while Peter gets his computer ready.
So what's the mechanism for exclusion from the nucleolus? Maybe I missed that.
Oh, you didn't miss that because I didn't tell you that. Sorry. I'll use this microphone for now.
You can use this one.
Okay, perfect. So a couple of years ago, what we did, we asked exactly this question of, how does, let's say, condensate X keep its components together from condensate Y? And the challenge that we came up with was the speckles versus the nucleolus, right? And it turns out that speckle proteins are defined by intrinsically disordered regions that are essentially RD or RS repeats. So whereas poly-Q, and in particular the N17, they like to interact with RD repeats, with RS repeats, and so on. So in fact, PQBP, which is a poly-Q interacting protein, loves RD repeats. But there is a sort of considerably weaker interaction with the building blocks of the nucleoli, which we've recently discovered are just blocks of lysines and blocks of glutamates, right? And so in the IDRs. And so just the same specificity that allows for separation into the speckle versus the nucleolus is what Huntington appears to be hijacking as a way to sort of keep away from the nucleolus, but it's happy going into speckles. It's happy going into paraspeckles based on this code. Yeah.
Okay, thank you, Rohit.